Hi, Merrick, Marty here. Can you hear me okay?
Yeah, Marty, how you going?
Yeah, really good. Thanks for joining us.
Yeah, no worries. Pity I'm not on the boat. Last dive, very exciting. And
Yeah,
down 2,000 metres.
last dive, 2,000 metres. Yep, we would have loved to have had you on board with us.
Oh, well, COVID, etc. Couldn't get it there. Yeah.
So while you've been sorting out your audio connection and what have you, I'm not sure how much of the video you've actually been watching, but if you've seen this rather unusual specimen that we've just collected.
No, I just saw it go into the quiver. That was all I saw. Last I saw you looking for a spider, but then the feed dropped out. So.
Everyone here is scratching our heads saying, what is that? Anyway, safely tucked away into one of the quivers, and
looks like a vaguely like an octocoral to me, but anyway.
Yeah, yeah.
Unless there was a bunch of zoanthids attached to it, which I also have collaborators in Japan who'd like to look at that. Yeah. But hopefully find some more stuff on the bottom here where it's nice and deep.
Yeah, nice and deep today. We don't have a lot of time. We can't really do much of what we would normally do, which would be an exploratory dive where we go on a bit of a tour. But we're so deep today and we've already done the midwater plankton part of the survey, so we really can't linger longer. We need to kind of stick to our transect and make our way towards what we hope will be quite a hard rock outcrop, the face of this plunge pool that is about a 650 metre high cliff that we'll be ascending up. So hopefully, obviously we're keeping our eyes out for carnivorous sponges, which is your specialty, and we're just on a soft sediment substrate at the moment, but ahead of us we should fairly soon come to the base of the cliff and we'll start making our ascent.
Great, yeah, well, hopefully we'll whiz past some carnivorous sponges and grab a couple on the way up. A lot of them occur on soft sediments as well as hard sediments.
Yep.
Rocks and things that I like to sit on, yeah.
Hey Merrick, it's Rob Beeman here. Can you hear me?
Hey Rob, how you going? Yeah, I can hear you very well.
Yeah, good. We're trying to squeeze a lot into this dive today. So we've had Dhugal on most of the morning, and now we're on the seafloor. It's 2,054 metres deep, 2.3 degrees C. We've taken out, well, look at some big blocks here. It's quite a lumpy seafloor. We're going to have to continue moving. We've got quite a high vertical height to reach today, at least 600 metres vertical height we're trying to get in a couple of hours. So we'll do some sampling, brief sampling, as long as it doesn't take too much time, and then we'll just continue upslope. This is a big block here, it's fantastic. Big anemone on it. It looks like we've got probably a bamboo coral, or we'll see if we can see the bamboo coral nodes. Beautiful anemones here.
That's good. I'm sure you'd love to take the whole block, wouldn't you, Rob? There's some good geology in there and fantastic fauna on the surface.
Hey Merrick, it's Vicky here. I don't think we've spoken before.
No, Vicky, pleased to meet you.
No, yeah, you too. This is quite an unusual lot of rocks we've found down here. And
So it's
we've got
perfect habitat for these guys.
yeah, we've got bamboo coral here.
Yep, you can see the nodes now down near the base. There are these sections and they've got these darker internodes. So we're going to try and get a sample of this for the quiver to be flash frozen, but we just need to snap a little bit off the end and put it in our quiver, and then we'll continue on our way.
It's quite an interesting field of anemones here.
So we think these rocks here are probably originally calcitic, so limestone. All the deep rocks that we found in this area have all been some form of limestone. Calcareous being possibly shallow water corals or other calcareous organisms like forams, but it's very, very hard rock. It's clearly quite old, probably Miocene in age, at a time when the first coral reefs developed up in the far, what's now the far northern Great Barrier Reef. But if we go back, say 20 million years or so, when the Australian plates first moved into these warmer tropical waters, we're looking at a big carbonate or calcium carbonate province that has left these, it's like a deep ledge and obviously parts of it have broken off, and we're looking at this sort of boulder field. We're at the base of what looks like an ancient waterfall. So if you think of a waterfall on land, there's always lots of rocks and things, boulders at the bottom of the waterfall, and I think that's what you know, you can imagine what this now drowned landscape is. Yeah, so just definitely sedimentary rocks, but still very, very hard, as we've discovered. You know, just trying to break a piece off has been quite challenging. So we're just prepping the quiver here, and just to remind people the two ways that we're preserving samples. Most of our samples are going into those bio boxes, and they just get preserved with ethanol, and they preserve the skeleton quite well. But we are working with the Sanger Foundation and the Smithsonian Institute through Jeremy Horowitz and Tom Bridge to do full genomic testing on these deep marine animals, including the symbionts, the animals that live in amongst the skeleton and flesh of these living animals. And so the Sanger Foundation's aim is to do full genome test sequences of these animals, and to do that we have to preserve them very carefully, and we have liquid nitrogen on board. So we're just going to see if we can snap a piece off this bamboo coral and slot it into the quiver. When it comes back on deck, we will flash freeze it with our liquid nitrogen. We got something swimming towards us. Oh, it's a fish. We got a large fish swimming towards us. Yeah, this crazy fish coming in. We just saw the shadow, and it was like, oh, is this like an octopus or something? But it's one of these deep, these very strange deep fish. So I hope we got our fish army helping us here.
I hope it gets close enough that we can see it without having to move.
Okay, we'll just get this in the quiver and we'll have a closer look at this fish. And he's saying it's a cusk eel. Yeah, thanks, Glenn. We're also joined by John Pognoski and Will White from CSIRO in Hobart. They're our official fish taxonomists joining us on this expedition. But of course, we're taking advice from everybody ashore. We're very pleased to have you joining us on this dive. Myself, I'm a marine geologist, and Vicky, you're a Southern Ocean expert.
Yeah, so, well, Southern Ocean person. Yeah, I study radiolaria down in the Southern Ocean, looking at using them to reconstruct past ocean conditions, particularly over the last glacial cycle.
Yeah, and speaking of Southern Ocean, I mean, this temperature, it's 2.2 degrees Celsius. It's very cold.
Yeah, so this water originated in the Southern Ocean, and it comes up here and it brings with it the rather cold temperatures.
Just had an enemy, the fish is having a bit of a swim around the bio boxes in the other camera. You still with us there, Merrick?
Yeah, I am, Rob. How you going?
Yeah, okay, so yeah, we're just putting the quiver away and then we'll get going. But we did see some of these strange Christmas tree-like siliceous sponges on the seafloor, so we'll be able to stop and probably sample them since they come into view.
Yeah, sounds good. I was just trying to see if the camera can look slightly to the left between the brittle star and the bamboo coral, whether that was a shadow or a light cream kind of a sponge on them.
Yeah, just to the left of this brittle star.
To the left of the brittle star, yeah. Starting to look like a smaller one of the bamboo corals, but
Okay, so not sponge then.
I don't think so, but I might quite get a good focus on it yet. But it's about the size we're looking for. Unfortunately, they're not huge big things. That's why they weren't discovered until the 90s, really.
Okay, no worries. Yeah, we just thought that might be a sponge for your benefit, but we're going to get going now. We've got about 600 metre vertical climb in front of us, so we'll just we'll stop, of course, if we see something amazing, but we need to get a bit of a move along here.
So Rob, just remind us again what the goal is for this this section here.
Well, number one is the curiosity. What would be living at the base of one of these big plunge pools? So this plunge pool is a big scale feature. Imagine a waterfall on the... Oh, there's a great tripod fish right in front of us. There's a lot happening in front of us here. I'll get to context descriptions in a little bit, but let's just stop for the animals, eh? Bathypteroides. Sorry, Bathypteroides. Have I got that right there, John? Yeah, quite amazing. We have seen a few of these on our previous dives. I never get tired of looking at them. I'm just curious, are we looking at spines or rays that that hold the fish off the ground? Like, are they hard spines, or are they just very, very soft rays holding the animal up? And I'm guessing the pectoral fins on the side, they're part of this detection of animals coming past, and then I just grab them, I guess, as they detect something swimming by them.
It's pronounced Bathyopterus for those who are interested.
Yeah, thanks, John. Modified rays that are slightly spine-like. Very, very handsome fish, and here's some kind of rat tail fish coming in the background. So we got a lovely, really quite a large cold water coral here on the right. You might be able to get a closer look at it, and a very white, bleached white, possibly squat lobster, just to the left underneath this rock. Yeah, so you asked me before there, Vicky. I mean, this is, imagine a waterfall, the base of a waterfall, and there's a lot going on here. And, you know, we've got lots of boulders, and these boulders are very likely limestone that have broken off through who knows what geological forces have broken these off, perhaps even earthquakes. And they've tumbled down slope, and now they're just lying as a scattered bunch of boulders down in the base of this plunge pool. And it's about 800 metres wide, and it's got about a 600-metre vertical cliff that we're going to try and climb up today. So we're really at the base of this sidewalls, and these sidewalls are all, we're seeing just big tumbled boulders. These are sedimentary rocks, but very hard, as we've found out. Wow, look at this jumbled landscape. And of course, when sedimentary rocks break, they tend to break along the weaker sedimentary layers, the strata, and so we end up with this kind of flat pancake look. You can actually see some of the layering in these rocks, but boy, can you imagine you think about the geological forces you need to actually break off such a tumbled mass of boulders at this scale.
I guess when you think about the size of the waterfall that's at the top of it, though, the scale of it is enormous, and the power of the water that would have been up at the top. But yeah, as you said, maybe faulting and earthquakes could have brought some of these down as well.
Just a cusk here on the left, just coming into view now. Obviously attracted by the lights. There's a second one that's drifted towards us. Boy, aren't they a strange looking fish.
Rob, we've had a question on YouTube about why the rocks are shaped the way they are. They're quite block shaped, and there's a question about that.
Yeah, sure. I mean, sedimentary rocks, they build up layer upon layer, and typically you have, weak layers, which might be originally mud, and then you might get sand building on top of that, and so you get this kind of wedding cake layer effect. Each component of it will have a different strength, and so when there's any kind of pressure or shear, could even be like an earthquake shaking, then they crack along the weaker horizontal layers, and you know, are fractured down whatever joints and other weak layers that are cut through the rock, and so you end up with this very blocky shape. When sandstone, for instance, is broken up, it tends to shear mostly along these horizontal layers. Because it's not horizontal now, we're just looking at individual layered rocks. I mean, look at this one on the right, really big flat layer, it just tumbled on each other. It's really incredible. But of course, there's actually some interesting marine life in amongst it all. Lots of nooks and crannies. We've seen some cusk eels already. Merrick, I'm not sure if you can see, it looks like a siliceous sponge. We're just going to zoom in closer to it.
Yeah, Rob, it does look like a glass sponge here as well. Yeah. Yeah, I'll play with that one.
If you're not going to sample this
under
one,
a microscope.
yeah, so you have to let us know if we're going to sample, because we really need to keep moving along here. You know, you have to decide fairly quick whether it's sampling or not. Otherwise, we can keep going.
Yeah, well, how many quivers you got left? I thought I was trying to reserve them for ones that are really good.
We got plenty of room, so it's your call, Merrick. You just say what you want, and we'll collect. You know, this opportunity may never happen again in our lifetime, so let's make use of it while we have.
So if you look at small things, see that little white pendula hanging off the edge? That's what more like my target species look like.
Merrick, we've got 6 quivers left, and we do have loads of room in the bio boxes, but in terms of the quivers, there's 6 left.
Okay, that's all right. I'm happy to go to the bio box. Yeah, what's that guy there looking? It's really good. Interesting.
Apologies, Merrick. It's actually 4 quivers left.
That's all right. I'm happy for samples to be lumped on top of each other if we can get it. Like you say, it's a once in a lifetime to be able to get out there.
I think we might need the avalancheometers today.
Yeah, gee whiz.
Move one of these.
Look, but which one do you pull out, or do they all just tumble down? It's that coral Jenga again. Deep sea Jenga.
Yeah, if it's possible to grab one of those sponges that look like a lollipop at the moment, that'd be fantastic. If we put it in there.
All right, we're going to try for this one.
Do you want that in a quiver, Merrick?
Yeah, put in a quiver if you can, yeah. Since we've got 4 left.
Merrick, did you want it flash frozen or in ethanol?
Ethanol's fine, yep.
Ethanol? Okay, thanks.
Flash frozen, yeah.
And we got ourselves a rock, because we love our rocks here. We weren't going to we're not even considering breaking this off. This would be so hard. We've tried before, we just end up polishing it. So this will be fine. We can actually break it here on board the Falkor, so it's just a lot easier to do the snapping and breaking once it's back on the ship.
If it's possible to get one of those lollipops before we get too far away, possible?
Yeah, sure. No, we haven't lost it. We'll just tuck this bit of rock away, and we'll have another go at that sponge.
Yeah, no worries. Thank you. Oh, sorry, I get too excited about the carnivorous sponges. I only started working on them a couple of years ago, but the great thing I love about them is every time you get one, it's a brand new species.
Yeah, Merrick, have you got any of the sponges from the previous expedition? They're down in Brisbane already?
They are in Brisbane, but I haven't had a chance to look under the microscope. Unfortunately, the museum here has been, they use the COVID as a good time to renovate the whole place. So we were basically kicked out of our labs and offices for the last 9 months. So I only just got into our labs 2 weeks ago, so I can just start working again. So, but I'm dying to, believe me. It's nice when you get work you're excited about doing, isn't it? It's one of the things I love about science, just being able to do new stuff.
Yeah, but you know, just to be able to see the environment in which they're collected. I mean, you've talked a lot about the early expeditions. It's all like rock dredges, and you kind of put it down the seafloor and hope to get something. But to see them connected and attached to the their surrounding environment is just a whole other a level of excitement.
Yeah, that's exactly right. Actually, seeing what they look like underwater for the first time is fantastic. A lot of them will lose their shape on collection, so.
Got a sea cucumber just above this. You know, I always think of sea cucumbers as in the soft sediment environment. This one's just clinging to the side of a rock.
Yeah, that's right. And because they want to be away from a lot of the sort of snow, a lot of the times if you're attached to a rock or something, usually in a better position. So you don't get smothered by snow and you can spend all the time trying to catch things. Right.
Yeah, Vicky's just pointing out some of the cleaner faces of the rocks. You can see the strata, the different layering in the rocks. So, you know, that just explains, it shows much better the sedimentary, I guess, shape of the rocks as they appear here, because these are all just tumbled down in a great chaotic mass. But in fact, when they were first laid down as calcium carbonate, they would be in nice horizontal layers.
Yeah. Perfect catch, guys. That's great. And it's even got the hold fast where it was attached to the rock, which is great. As I was saying before, with things like rock dredges and that, usually all that sort of part of the specimen is all lost. And sometimes it's very critical. It's got different type of spicules in that part of the sponge, which lets you tell it what type of they are. So that's great. Great work, guys.
So you're saying the whole animal is just as important as the working part, the big tulip part of the sponge. Is that right, Mary?
Yeah, that's exactly right. Some of them have really distinctive spicules, which are like ones shaped like little mesh, like jigsaw pieces in the basal holdfast there that are in no other part of it. And that's how you can tell species and sometimes even genera apart. Yeah, sponges are pretty cool. One thing I love about sponges, they're a bit like stem cells, that they can direct different parts of them to become different organelles, if you like. So sometimes, particularly with carnivorous sponges, when they actually capture something, they'll actually tell that particular cells, okay, you've got to be a digestive organ now, so they can break it down. And then later on, it'll tell it to organise it to become another cell. As yet, no one knows how this is done. It's another bit of science we don't know.
All right, Merrick, your call. You just tell us to stop if you see something really interesting. We're happy to take samples, but we're just going to continue cruising up slope. So we've just started at the very base of this plunge pool, and it's an even more dramatic environment than I expected. Boy, like I was thinking it would just be a kind of a sidewall that we've seen in submarine canyons, which are kind of muddy but steep. But this is it's quite surprising, really, just how dramatic this landscape is. Anyway, you call it, and we'll stop. I'm seeing a xenophyrophore down here on the bottom left. It's really amazing. Foramnifera make these quite spectacular sort of golf ball or softball sized tests on the sea. Oh, big spider crab coming into view. Just stop there, Chris. It's just underneath us. There's lots of animals crawling around in the nooks and crannies here. Just got a glimpse of it. We're going to see if we can angle it down. There it is. It's just under, just on the right of centre. There we go. Big spider crab. Definitely dinner plate sized. All right. Thanks very much, Chris.
I think you're going to get in trouble if you keep stopping us like that.
Yeah, it's true. We have to keep going. I've already been to the bridge once to say, oh, we might be a little delayed. Anyway, we'll keep moving. Boy, so many things to see here. It's great. And the geology is really spectacular. Yeah, these tall colonies, they're likely to be bamboo corals, like the one we sampled when we first landed. And we got these really interesting anemones popping into view. They're actually, you can see them under most of these rocks here, and a few sea cucumbers. Yeah, feather star just sitting there. I'm on the lookout for these stalked feather stars. These are the sea lilies. We may see them. They're certainly, we've seen them in these depths before.
I'm quite surprised how much life there is down here, sitting on the rocks.
Looks like we've got sponge here. It's not like... Can you see that one, Merrick? It's just disappearing up there. Oh, we've got another cusk eel coming out.
Yeah, I can see it sliding past.
God, it's so strange down here. These very... The fish life is so strange. It is alien, isn't it? Alien-like there, Vicky.
It is quite alien.
A rag cusk. Thanks very much. I see something white over here on the right-hand side here. Just looks like plastic.
Yeah, it looks like rubbish.
Well, it wouldn't be the first rubbish we've seen here. Down in, where was it, Rodder Canyon, we actually saw quite a bit of rubbish. Remember, this is part of far northern Great Barrier Reef where very few people live, so we can't really point the finger at communities close to the coast because there really aren't any. This would have to have been brought here by ocean currents, surface currents from, you know, much further away. There we go, Sebastian doing the right thing, giving a bit of a letter collection. And looks like it's got writing on it. Yeah, it's definitely plastic. Yeah, for sure. Don't recognise that. Looks like a logo or some writing. But you can see it's I mean, it's hardly degraded, is it there, Vicky? It's actually quite intact.
Yeah.
Bag of rice, John's saying.
Yeah, bag of rice. That's a shame. It's all the way down here. But not surprising, I did worked on some sediments in Antarctica, Prids Bay, and I was finding microplastics in the sediment down there. So yeah, it's unfortunately not very surprising.
Yeah, and I first became really aware of it when I did a voyage on the Southern Surveyor, so Australia's Marine National Facility vessel, back in 2000 and 12, we went from Brisbane to Fiji, and about every degree of longitude we stopped and did a very fine microplastics trawls, only about 5 minutes, in the top 10 centimetres of water. So it was pretty brief. And we just did that all the way across to Fiji. And every single trawl had microplastics in it, so just drifting around that surface waters. But of course the bigger pieces end up becoming waterlogged and down here on the seafloor.
Yeah, unfortunately this is the kind of plastic that damages the turtles and whales and the bigger marine life.
Okay, looks like we're running out of boulders here. It's not that big dramatic boulder landscape. Just the insert shows the 3D map that we've been producing over the past few weeks, and we're in what we call a plunge pool at the mouth of Wreck Bay. That's this very large horseshoe-shaped bay that's about 20 kilometres wide, so it's quite a big bay. And the plunge pool is, well, it's the second of 2 plunge pools. Imagine a great waterfall carved environment when this land was, you know, the long geological past was up above the sea surface. So it was exposed to the air, and fresh water would have carved this landscape. And I would say we're looking at boulders that have been knocked or broken off from the edges and tumbled down slope. So we're at the very bottom of this. It's about a 500 metre high sidewall to this plunge pool, and we've just started our climb up. And a shout out to my brother Greg, who's watching from Kiora and Sunshine Coast. Welcome aboard, Greg.
And I think my sister is watching as well, Kerry. So, hi Kerry, hi Will, hi Wynn, Glenn and Millie.
We just went over Anthomastus. This is a really interesting soft coral. It's a Gorgon's head soft coral. We're going to see more of these, obviously.
So it's at 2.3 degrees here, and when we were coming down it was 2.3 degrees for quite a way. It's going to be very, very cold, probably right to the top of the waterfall here.
Yeah, so we got sea cucumber just passing underneath us. Got a big squat lobster sitting on the rock centre right. and Vicky, what do you make of these? They look like big tubes underneath on the underside of rocks.
Yeah, it's hard to tell what they are, whether they're polychaete tubes. They're very large for polychaete tubes.
We do get worm snails at this depth.
Yeah.
You know, they're gastropods, you know, they're snails, but they make these long, fat, calcareous tubes. So I wonder if that's what we're looking at.
Yeah, possibly.
It's more anemones at the top of the screen. Yeah, this is, I guess the beauty of this sedimentary rock, when it fractures, you have these very big flat ledges as they tip over on each other. So lots happening underneath these ledges. just having a look in under this rock here, there, Merrick, it does look sponge-like with all those filaments hanging out. It looks a bit awkward to pull it out, though. It's kind of tucked away under that rock.
It's got filaments, yeah, it's good for me. It's a bit hard to get to, doesn't it?
What do you think? Is this one that you think we should try and collect? Because the guys can try for it.
Oh, I can have a closer look at it. I haven't got a very good image of it at the moment.
So do you think it's alive there, Merrick, or is this just the spicules, exposed spicules of a dead sponge?
I don't think it'll be a dead sponge, but I'm not even sure it's a sponge at the moment. Yeah. If that one's difficult, let's move on. Or, yeah, I'm not even sure it's a sponge, that one.
We can try and grab it for you. We'd probably put it in the bio box.
Yeah, grab it and put it in the bio box. Actually, you see the filament above it hanging down? That's actually even more interesting to me, because that's single filament.
Roger that.
And that's just for ethanol, Merrick?
Yes, just put everything in ethanol's fine. Thank you, Vicky.
Whoop, just slipped out of our fingers here, so just trying again.
Yeah. Unfortunately, that's a problem with my ones. They're so small. In order to see them, you have to go really slowly and really zoom in to see them, which is why, you know, they weren't really discovered until the 90s. All this time. Great one. Thank you.
Just pulling away from the rocks here while we tuck away the sample, and then we'll continue up slope.
Great work. Very exciting.
So, Merrick, I take it you'll only be able to look at the spicules after this has been preserved. Like, there's not much you can use from the flesh, or what is it in these siliceous sponges? What's the non-silica part of it?
Well, there's other proteins in there. There's the spicules do two things. One is they provide the structure to support it, and they also provide protection. In this case, they are used to capture the other things, but there's other proteins in there. Collagen is one of the main ones that you'll find making up the rest of the body. So it doesn't really matter. It's easy to get DNA from the rest of it anyway. It's not like the black corals where you only get the flesh on the outside, because the flesh is inside all of the sponges. Sometimes they have spicules as armour, but usually it's a centre like a like a bone skeleton, if you like to think of, in humans or something, and ones on the outside. So there's no problem about not having enough tissue.
Yeah, we got a nice cup coral underneath this big boulder in front of us, and it looks like probably bamboo coral. I got a good question here from Radigar. Why is there no ferromanganese incrustation on the rocks? And that is a good question. It's not as dark as I'd expected. Typically, rocks, when they're exposed to the seawater for really long periods of time, and even millions of years, have this very, very dark crust on it. We've seen this over in like Osprey Reef is a good example. All the rocks we saw there, very, very black, and in some parts of the coral sea where I've sampled, you can have this ferromanganese oxide build up like this huge crust, centimeters thick. But here we're not seeing it so much. Now this might speak to how recent, in geological terms, this rockfall may have happened. So that really we're not talking about rocks that have been sitting here exposed for very long. Again, it's a bit of a question as to how long. Perhaps that rock that we sample earlier will give us a clue as to when, the age of these rocks. But just the fact that it's not covered in ferromanganese oxide just says to me that they've been tumbled down in more recent geological time, as opposed to being exposed on the sea floor for millions and millions of years without any disturbance. We're just going to boop this one for science. Okay, boop for science. Oh, a little bit of reaction. All right, might try its neighbour. So, quite different animals here. One's a sand-dwelling anemone, the other one's completely attached to a rock. Let's see what we get here.
Bit of an anticlimax boop though, one. Yeah, exactly. At 2 degrees, I don't think I'd be responding very quickly.
Yeah, that's right. Everything moves slowly down here. So, it's I guess about energy and things like that. These animals don't have to move much at all. They just sit there quietly and growing slowly. We know that the cold water corals take, hundreds of years to grow in places. So everything moves, slowly. So it's probably no surprise the tentacles didn't pull in as quickly as, say, shallow water anemones.
What are these big sponges here? Merrick, do you know what that is? Almost looks like a lily. Just moving over to it now.
It's hard to tell if it's a glass sponge or a rock sponge from here. The rock sponges are probably corallistes, and they've got amazing spicules. And this is one that's got these 3D matrices. It's just like 3D jigsaw puzzles all wedged together, and it makes up the whole solid thing. So it's solid glass. Possibly corallistes, yeah.
And a shout
like some of the glass ones, you pull up are probably a bit softer. Some of the glass sponges you probably saw, like the sea lilies, are a bit softer. You pull them up and they're made of glass, but it's a fine like mesh. And other ones that it's like solid as a rock.
Just a shout out to my wife Diane, who's just joined us on Facebook. So hello Diane, I'll be home in a few days. This is our last dive today, and we'll be heading back to Cairns tonight.
You've had pretty good weather the whole way, Rob.
Yeah, I did get seasick on the first day, but we've been really lucky. It's very nice conditions up here. The water's warm, the sun's shining, got a light breeze. Yesterday was like a swimming pool on the Great Barrier Reef. Merrick, do you want this sponge? Otherwise we'll keep moving.
If it's easy, grab it. It's not a high priority, that one. It looks a bit hard to get in there.
Okay, we'll continue up. Yeah, I think we've got a sea lily here. I mentioned these before. If this is one of these sea lilies, it's a stalked crinoid.
Have you collected any stalked crinoids yet, Rob?
No, we haven't. So, you know, our focus has mainly been on sponges and corals, and our permit does allow us to collect other invertebrates, but we're circumspect about how we do that. So, you know, for this dive, our focus is really on the sponges for your benefit.
Thanks, Rob. Pity it's so hit and miss with my sponges.
We've had quite a few objectives for this dive today, so we wanted to do the Dhugal dive, and we managed to stick to time there. We've gotten to the bottom. Weren't sure we would fit everything in, but as we're disciplined, we'll stay on track.
Yeah, but what a landscape to explore. We're enjoying looking at the rocks, the different strata here. You know, we're just remarking how sharp-edged all these are. You know, as if someone's hit these rocks with a giant hammer, and they've split along the horizontal strata, but also vertically. But this isn't unusual in any kind of rock that's weathered over time. You get faults in it, and so they can, you know, when there's any kind of forces breaking it up, they shear along whatever the weak sediment plane is, and also across the fault lines, you know, the cracks, and so you can end up quite easily with big sharp-edged boulders as we've got here. But it's still remarkable to see, you know, such large boulders all tumble down on each other.
So Rob, what's the pressure down here? How many atmospheres are we experiencing on the ROV?
Just over 200 bar, 200 atmospheres. So if you are a scuba diver, you typically pump your air pressure up to this same pressure, right? So how I remember it is you look at the depth in meters and you divide it by 10. So we're over 2,000 meters, we're in about 200 bar, and I'm a scuba diver, and your air pressure is usually pumped up to 200 bar or atmosphere. So there's a lot of pressure, and of course, anything could implode. Here we had a lovely stalked crinoid sea lily right in front of us. We're just drifting over it.
We just got a report from just under 3,000 psi apparently.
Yeah, 200 bar, about 3,000 psi. So some serious pressure here. All right, just reaching out to our scientists ashore, giving us some advice on some of the coldwater corals to collect. So we may, if we see a bamboo coral, we'll try and get that in a quiver so we can flash freeze it. There's a nice cup coral right in the center of the screen. We're not going to collect it, but it's a classic cup coral with all its tentacles out. Yeah, Merrick, we're just moving over to this bowl that's got these bamboo coral colonies hanging off it. More for the genome project. If they've got any animals like amphipods or what have you attached to the tip, we'll see if we can break a piece off. Otherwise, if we can't see it, we'll just continue flying up slope.
Yeah, that's good, Rob. Yeah.
Okay, we may have a candidate here. This is a bamboo coral, and we've got like perhaps a little squat lobster sitting in amongst it. Lovely sea lily in the background there. This might be one that we can sample for our quiver. Yeah, good question. Does animal life suffer from the bends? Well, certainly there are fish with swim bladders, mainly in the shallower waters. They have swim bladders to help them keep them neutrally buoyant at whatever depth they're in. You know, if you catch them on a fishing line and pull them up too fast, the swim bladder really expands and can kill them. Down here it's a different story though. I'm not sure if the fish here have swim bladders. Maybe John or Will can explain that one. But you know, when we're bringing animals, these are samples from these deeper depths. Not many of them are actually alive by the time they get to the surface. You know, this is something that we consider. We are not just willy-nilly collecting samples for sample's sake. We have a real decision-making process on board about what we intend to collect and why we are collecting them. So we only collect within the limits of our permits, and if we already have a sample for a particular need, we don't collect any more than we need to do that, just for the science. Right, this is one of these samples going to be flash frozen. So we'll put them in the quiver, and when it gets up on deck, we'll put it into liquid nitrogen. Yeah, and thanks, John. John's just saying that some of these really deep sea fish also have swim bladders, so they could suffer from the bends, you know, reverse bends as they come up to shallow waters where their swim bladders are like a little balloon with gas inside, and they would expand too much and kill them. But you know, we're not collecting fish here. We're just looking at them and admiring them.
So some of those swim bladders are much possibly smaller than the shallow water species. They're not as big. They do perform the same task, but yeah, no collecting of those today. I don't believe we've got a permit to do any vertebrates at all.
No, that's right. Sorry, John and Will, no fish collecting here, but certainly the vision's pretty good, and that should help for any post-voyage analysis. And leg 3, which happens after we arrive back in Cairns, leg 3 is going to take the Falkor through to 17th of November. No, the ROV won't be used. It's only mapping, and there's a few other things happening through that as well. We're going to do a lot of some more CTD, conductivity, temperature, depth profiler, sampling down in the southern Great Barrier Reef, and then the ship's heading out into the Tasmantid seamounts, which is in the Coral Sea Marine Park. The Tasmanian seamounts are a chain of extinct volcanoes, and we've got some interesting tasks there. So the Falkor will be mapping around the steeper flanks of these extinct volcanoes. They're within Australian waters, and some of these extinct volcanoes actually have coral reefs on top of them, including islands and sand cays. They're some of the most isolated sand cays islands in all of Australia's marine estate, hundreds of kilometres away from the mainland, and they're very important for seabirds and turtles and things like that. So we're going to try and get the Falkor team to go on there to help Parks Australia get a snapshot of marine debris, or if there's any invasive species on there like ants or things like that. That would really disrupt the ecology of these really small little dots in the ocean. We'll have our media correspondent taking lots of photos, but it'll be quite an interesting addition to the normal expedition that we originally started planning for about a month or so ago.
You've seen some of those islands before, haven't you, Rob?
Yeah, that's right. I was in the Navy and worked on a vessel that had a helicopter, and we were dropped off on some of these remote islands to do geodetic work. We're basically setting up geodetic GPS to accurately position where the islands were. Now that might sound funny in this day and age, but you have to remember back, well, pre-GPS, a lot of these islands were only marked by astronavigation, where people had used sun sights and to work out exactly where they were in geographic space. And so going on the ship was a chance to put these very accurate GPS on the middle of the island, and so they flew us out by helicopter and left us there for marooned. Quite amazing islands, really. They're not big, you can walk around them quite easily, but you know, just absolutely covered in seabirds, and you'd see turtles and sharks swimming in the shallow waters. And so you guys are going to have all of that fun ahead of you in the next few weeks.
So I have to ask, how accurate was were the maps, previous maps, once you got the GPS points?
Yeah, so rule of thumb with that kind of astro-style navigation is, you know, you can pretty accurately find yourself on Earth within about a nautical mile or so. So they weren't too far out, but of course, as satellite imagery got better and better, and people needed to, you know, we needed to improve our charts. What we did was we actually flew onto the island and we set a benchmark down there, so that you may even see that, might even still be standing. So we actually took cement and put a big metal benchmark into the middle of the island, and that's kind of a solid bit of a fixture point from which you can get your latitude and longitude, and that hopefully they'll still be there.
Well, I hope I'm one of the people that are picked to go ashore that day so I can find your benchmark.
Yeah, good, Vicky. So, but you know, it's just fascinating places, and there's also shipwrecks on them, some of them quite historic shipwrecks. So that will be a part of this leg 3, as well as the mapping that we've got planned around the steeper flanks.
Yeah, I think it might be time to keep moving now. We're still at almost 2,020 metres.
Yeah, and we've
still
got
a long way to go.
That's right. It's about a 600-metre climb is what we planned, and we just need to just keep on moving upslope. So again, Merrick, please sing out if you think there's a sponge that we should stop and collect. We're happy to do that. Otherwise, we're just going to do this lovely drift over this quite spectacular landscape.
Yeah, don't worry, Rob. I'll sing out when I can see something worth collecting. Yeah, it's just great to watch.
And the sunset view shows where we are. We've dived into the centre of this, what we think is a giant waterfall carved landscape that would have been exposed at above the sea, you know, probably millions and millions of years ago. The whole edge of Australia has now warped downwards, subsiding. Now down, we're at 2,000 metres deep. So this rock probably was shallow water if it's limestone, but now millions of years later, it's 2 kilometres down, and it's been carved. It's like almost big valley. And we're just climbing up the side of the sidewalls. And you can see how this is just the 3D depth model, how steep it is, but in reality, it looks just far steeper. I actually thought it was just all mud and sand, you know, like we've seen in some of these other canyon environments. But, you know, to see these jumbled boulders like this is really quite remarkable. We're saying cat shark is coming into view. Cat shark behind us. We're just going to spin around here. Will White, if you're watching, we're just trying to find a cat shark that came into view. Because we've got 10 cameras on us, we can look 360. What you get is the science camera most of the time, but we're just spinning around now to see if we can pick it up. Yeah, we're looking at the other cameras here. We're just trying to spin it around in the water, spin SuBastian around the water column here to look at this cat shark. So if Will White's watching, if you go to the Schmidt Ocean Institute website, the latest cruise blog is a critter feature by Will White, one of our fish taxonomists, and he's written about the cat shark that we saw earlier. Here we go, coming into view now. No, it's another cuskeel. No, we've lost, looks like we may have lost that cat shark.
So we've got a wall on the side here. It looks like it goes for quite a way. It's very layered.
Oh yeah, I mean this is classic sedimentary rock here. You can see where all these boulders have come from. This is the outcrop, the laid outcrop. We're going to see if we can grab all this rock here. Boy, geez, look at that. It's just, yeah, the faults. So this is how we're getting these really sharp edges. You've not only got that horizontal layering, of course it's not horizontal now, it's all warped downwards, but there are these cracks and crevices broken through, and so this is how you end up with these really sharp cornered boulders just peeling off the edge. Now notice how black, how dark this rock is compared to the boulders down slope. So this would be a face that's been exposed for some time now. This is a black ferromanganese oxide coating. We're going to try and find a rock that we can pick up. I don't think we're going to break anything off, but if there's anything loose, what's good about this is we can see what layer it's at. We know that it's actually outcrop rock.
Sebastian's going to have a go at breaking it off.
Just having a bit of a poke around, see if there's anything that's sticking out we might be able to lever off. No, I think the rocks beat us this time. We're just going to see if we can find anything loose. We have tried this in the past, Merrick, and it's just too hard. We've ended up polishing the rock at times. So. We're going to try and grab a bit of rock before we head up onto that soft sediment. So just keep your eyes peeled for any loose small boulder or rock. That'll be perfect for us.
Looks like a candidate. Hey, good job.
Well done. Woo. Perfect. Perfect. Now we can actually break that here on the ship, and that's just exactly what we need.
I think the rock knows we're in a rush today. Might have a few of those tubes attached.
Yeah, we've seen these really large tubes. There's really large tubes, haven't we, Vicky? Curious as to what they are.
I might have to put those under the microscope when we get it back into the lab.
There we go, there's some there. That's a keeper. That's exactly what we're after, thank you.
Good job, Joe Rod and Cody. Thank you. Team Texas.
So Clint's asked about the water here. So just to put things in geological perspective, the rock that we're seeing here probably was made in shallower seas, so shallower, higher up in the water column, much longer ago. And then as sea levels fall, or this would have been exposed, then fresh water then carved the landscape into this waterfall style plunge pools. And then, to make it even more complex, the whole edge of Australia has been angled downwards, so the whole area here has subsided, and so it's now deep down and 2,000 meters down. So it's a little hard to get your head around the long geological time and processes that have acted on this landscape. But you know, you can only explain that the carving that we're seeing through fresh water, when all of this may have been pushed up and exposed to the air. But the actual rock itself is shallow limestone, most likely, and would have been made in shallower seas. Probably even, you know, during the Miocene, so 20 million years ago, when Australia first moved as a moving plate into warmer tropical seas. That's when we see the first turn on of limestone rock at the very far northern reaches of the Great Barrier Reef. So this is a very ancient landscape that's had a lot of different forces acting upon it. So it didn't form down here, it's simply the preserved remains of a carved landscape that is now subsided and now deep down, 2 kilometers down underwater. Oh, we've got something really interesting right in front of us, and yeah, we have seen these before. What are they? Merrick, they react. They close up like a balloon. They've got big tentacles at the end.
Oh, Rob, I think it's some sort of an enemy, but I'm not sure. Oh, it looks like a jellyfish there.
It's not a corallimorph, is it?
Well, corallimorphs are sort of basically a type of anemone, so it could be, yeah. We found quite a few of them on the deep sea trawls as well.
Okay, but we'll continue moving up slope. Still got a ways to go.
Hey, yeah.
This looks quite different now from what it was over 2,000 meters. We've now just got mud again.
Hey Merrick, we're just going to swap around here. Vicky and I are just having a break. We'll go on to the loggers, and we're going to bring our 2 students in who can introduce themselves. Okay, you ready?
Ooh, a flatworm? Scale.
Could you guys have a quick look under that shelf there?
Yeah.
Those ones hanging down, the little pendulous type things off the roof. There's some carnivorous sponges there.
So, hi Merrick. So can you describe it one more time?
Okay, go left where the black section is. There's tiny little, like a sliver coming down. It's got like a little disc on it. Down, down. The bottom one.
That one.
Okay, I'm having trouble seeing it on my screen.
Is it the one with like a little orange dot in the middle?
I can't see any orange dots, but
we're zooming in.
see the ones that. Yeah, those ones there. The little really white ones that have whole lot of filaments coming out of the centre.
Yeah. This one.
If we could grab some of them, that would be amazing. Yeah.
Do you think it's easy?
Oh, now I see the orange one. I can see the orange one as well. If you could possibly get all three, that would be fantastic.
All three of these in the frame right now. Okay. Can we grab and put in a bio box?
Yep. Oh, that looks fantastic. Yep. There we go.
All right.
Another 2 new species.
There are a lot of going on before Valerie and I can reintroduce us again and say hi to Merrick. We've been collecting sponge species for you for this like 2 voyage on Falkor, but we haven't officially said hi to each other.
Yeah, exactly. Hi, Merrick. It's nice to finally meet you.
Hello. Nice to meet you too. Yeah, I work at the Queensland Museum on sponges and ascidians and octocorals and lots of other groups as well. But at the moment, particularly on this expedition, carnivorous sponge is what I'm interested in, because there are just so many new species. It's fantastic. It's such a fun field to work in too.
I've never known there was carnivorous species before, so it is very interesting.
Yeah, well, people collected these a couple hundred years ago, but no one knew they were carnivorous, and it wasn't until someone in the 90s found some in a cave, shallow water cave or scuba diving depth in Europe, and they went, hang on, these are carnivorous. And they went back, and now people have gone back and looked at the old specimens and worked out, you're right, they don't have any aquiferous channels, they're not sucking in the water and filtering it. So, and that's really good from a describing point of view. They don't have 200-year-old wrong taxonomy, poorly described specimens to work from. You could start afresh, and so it's much faster. Yep, got one. Fantastic. Good work, guys. Plucking little daisies with a robotic arm,
Yeah, exactly. These actually also remind me of a couple dives ago. I'm sure that you watched it happen, but we collected those really, really small sponges, and we found like a whole field of them, and I know those were one of the target species that we were speaking to Jeremy about, and in just that small field, I think we must have found at least 2 or 3 different species.
Yeah, that was the one on top of the snow-filled boulder. Yeah, that was great. That was really good. So, chances are these are a different species again.
So until now, how many carnivorous sponges said we know already?
I'm trying to think. It's around about 100 between. I think it was about 150 last time, so I counted. I mean, I just described 17 new species from a previous expedition, so it goes up and up. Yeah. Great work, guys. And most important thing too is with this, we can get images of these things which have never really seen before. Because a lot of the time, as you probably heard me talk to Rob about, they're collected by a trawl or something, so they come up in this. They come up with all this mud and spicules from other things, parts of whale flesh and everything. And so when you've got something so tiny with fine little filaments and it gets encrusted in mud and every sediment, it loses a lot of its shape. So we get these fantastic photos. So brand new stuff.
Yeah, exactly. Woo! And as you said, even these samples that we're collecting right now, even though we're not trawling them, once we bring them in the lab, they already look completely different to when you see them in situ, like right now. So it is really useful to get those photos beforehand.
Yeah, that's right. It's stuck there, yeah. And if you think about it too, we're talking about 2-degree water. So any animal you bring up, even if the surface it's only 18 to 20 degrees at the surface there, these things are going to be parboiled by the time you get there. So they go through a big pressure differential, but probably more importantly, it's a lot of temperature differential as well.
I think the pilot's just trying to grab 2 more, so in case.
That'd be great, thanks.
Yeah.
Yeah, fantastic. They're so small, by the time I cut a bit out to do an SEM on it and cut a bit out for DNA, there's not much left of it. Great work, guys. Thank you. That made my week. Woohoo!
So I've actually got a question because Jeremy was also telling us describing all these carnivorous sponges. When we're saying describing a sponge, what are the features or characteristics you're trying to describe here?
Well, with most sponges, it's actually the silicon spicules what define basically family and genera and species. And you get everything from large spicules, which could be several millimeters long, but most of the ones we're interested in are probably about 7 or 8 micrometers long, and they'll have different shapes. So we use a lot of them. And we're also using molecular work at the moment to show that these particular spicule types are characteristic for the different genera and families, and that's what a bit of science is going on about. But also the morphology of the shapes, like you saw this one here, looks like a, if you like, a flattened lollipop with lots of filaments coming out in basically one plane only. So that's very different. So, you know, look at other ones. But there's classic ones that sorry.
No, sorry, go on.
There was classic ones that were described many years ago, and it had all these flattened sacs, and no one quite knew what they were for. And it wasn't until we had an ROV down with the camera down there, and someone went, "Oh, these are actually spheres that the sponge fills with a little bit of gas and liquid, and so it enables it to float off and holds it off the floor of the to be away from all the sediment." And so there's an example of another really different type of structure, really different shape. It's really characteristic. And yeah, and if we hadn't had cameras and ROVs down there, we wouldn't know what that was. But yeah, so it could use these spheres to hold it up rather than relying on all the silicon to hold it up. I mean, the good thing is there's lots of silicon down at this depth. There's 150 times more silicon in the water here than there is at the top of the ocean. That's one of the reasons why we have a lot of glass sponges down here as well. Oh, that's great, guys. Great work.
So, Marek, would you say that with sponges, since I guess right now you said for the carnivorous sponges there's only, over 100 species that are described, do they also have the same problem in taxonomy where some may be misclassified or are, classified in a way that you don't actually believe they're in the right genera, or is that not so much of a problem yet just because you're still discovering a lot of them?
Yeah. Yes, you're right in both ways. With sponge taxonomy, it changes a lot. I mean, the carnivorous ones, I've just decided to move these things into different genera, and some people probably have said another paper saying that's not right, but with sponges in general, yeah, it's no surprise at all. Suddenly, this family will move to a different order. That happens all the time in sponge taxonomy. Genera move around, families and all that. And if you've got to think about it, the whole idea of having genera and family and orders is really a human construct to make it easy for our pea brain bodies to be able to understand what's actually going on. It helps, sure, showing evolutionary trees and that. A lot of it's just so we can categorize things in ways that we can understand. And now that we've got molecular DNA, it's showing a lot of all these old sponge groups are totally wrong and moved around. So it's science in progress, I think.
Yeah, I guess that's a classic case, same as corals, it starts off as morphology and then it moves on to whether, you should define species by genetics. So it's I think one of those fields where taxonomy innately is quite a hard thing to get into, no matter, what species or what genera or what family you're looking at.
Oh, okay. You see, I don't know if your camera, but you see, oh no, well, that's a bamboo coral to the right there, the little feather type one. Yeah, sorry, but you're exactly right. Same sort of problem. And because a lot of these things were described hundreds of years ago, and there's a tiny dried up fragment in a museum in Europe that you may or may not be able to get hold of. You can't do DNA on that, so you've got to go back and recollect the whole things, prove it's the same thing, and then we can start moving ahead with DNA. And then, of course, with DNA, the problem is people are using small sequences, and then later on someone else will use a different sequence and probably get a different answer. And we could do whole genome sequencing. But then people will go, okay, what about post-transcriptional modification and all that as well? So, in another 10 years, there'll be a new technique we haven't even thought of, which will move us ahead.
So we've got 2 questions also on YouTube channel. I think the ROV team can help us to answer one of the questions. How do we calculate our current depth? Is it due to the pressure sensor? Yes, so that is one answer for that. A second answer, maybe Marty can help us to answer this. Did it go? There was one question about usually how strong the currents are when we're at the depths. Yeah.
Oh, this looks like an interesting sponge in front of us. Marek, do you care to comment? I don't know anything much about this. It looks quite new to me.
Looks like a glass sponge. Yeah, if it looks easy to grab, why don't we grab it? Yeah. I didn't guess a family on that. Yeah.
So this looks like an interesting glass sponge species, and we're going to collect this. And back to the current question, I don't know if you guys heard it, but our ROV team was saying usually when you go to deeper places, the current tends to be weaker, but apparently not in Australia. This is quite a special place, and we have the Antarctic current down below to influence the deep water here. Sometimes we can actually get really strong current in deep water as well.
Yeah, that looks good because that glass sponge from here anyway looks like there's one we keep collecting from these deep sea things, not from an ROV but from trawls and that. And every time we bring it up, it's been like what we call dead, because all the superfine spicules have been washed out and there's no living material. It's just the big spicular matrix is there, and you can't use that to tell even what order some of these sponges are. So, and we keep finding it, but we haven't found a live one. So because it's solid glass, we go, well, has it been dead for 10 years, 100 years? Or are these things like 1,000 years old and just hanging on like an old dead tree stump? And we still don't know what they are. So we keep hoping we'll find a live one, and that looks pretty good. And then we might be able to also do some DNA sequencing on it as well. So finally we'll be able to describe this thing properly, rather than just call it unknown. That's what I love about expeditions like this. Answer so many questions and ask so many more. But it's that beauty discovery of science.
Yeah, that's a fair
But I love
point
to
you
see
made, Marek. And I think what's so amazing is that we get to, as we said today, you know, it started off as a Dhugal dive and now it's a Marek dive, and we really get to discover so many things about so many different fields.
Yeah, I'm watching on YouTube's people's suggestions for names, so I'll keep that in mind if we describe a new one.
these rock formations, I can't comment much on them, but I'm sure that Rob and Marty would probably comment on how amazing you can see these clear stacks on top of each other. I don't really know why this happens. I don't know if anyone out there could comment on it, but I think so. It's like sedimentary, so it just deposits in layers. But yeah, it just makes this amazing formation. I guess when you know, as we're going up with it, and we get to see these beautiful layers, it's, you know, quite the difference from how we started the dive with those huge crumbling boulders. So, Merrick, I have a question about the range of sponges, sorry, that you've been collecting. Have you collected much at this depth, or is it, you know, one of the few times right now that we've been collecting samples at this depth?
It's probably the only time we've been collecting at this depth, at 2,000 metres with an ROV here in Australia. Some of the French people have done it in other parts of the world. But we've collected at 2,000 metres and 4,000 metres, but only probably about 3 times. There was a Challenger expedition in the late 1800s, and there was Sedaris exhibition in the 1970s, and there was investigator expedition back in 2017. So this will probably be the 4th time at these depths around Australia at all, but the first time with cameras and being able to pick them up. So that's fantastic. The rest was like trawls across the bottom and see what turns up. This is the first, yeah. 2,000 metres is good depth. I like that. I'm not concerned about anything above 500 metres, the shallow water, you know.
Yeah, exactly. So that's another Falkor first. I think that's very exciting.
It is very exciting. Just great to be able to have the camera down there and be able to manipulate and pick up things. Just spectacular. See things in their proper habitat.
Here on the YouTube channel, we're showing again an inset of topography map that we recently mapped.
Resume. Oh, it's gone.
I think we've been so lucky today for our final dive to get such a diverse dive with our mid-water dive in the beginning and then going so deep now, and just through looking at that map, it kind of takes me aback a little bit to see how we're going up this, you know, waterfall slope. And yeah, I'm just amazed. That's all I can really say. I'm so happy, I guess, for our last dive that we got to do something that was so diverse.
I remember like a couple of our dives when we dropped down, it's just sandy bottom, like really covered with fine sediments. And now this dive around this plunge pool that we have, rock formation, it's so huge.
Yeah. Is that a cool looking sponge there? Is that a sponge? Let me see closely on yours.
Is it the one next to the branched
Yeah,
octocoral?
it looks like a vase looking straight on it. If it's possible, could we grab that, stick it in the bio box, please?
Yeah, sure thing. We can put it in the bio box. So when you look at the sponge, it kind of looks like there's some algae growing on it, or is it just sediments that have deposited on it? Is that, I'm guessing it's not really like coral, where, you know, things depositing on it usually is a sign of death.
Yeah, no, I'd say it's lots of other things growing on top of it. You can get corals, you can have brittle stars, you can have worm tubes. In fact, a lot of the time we call them sponge hotels because they have so much epifauna attached to it. Oops. Okay, here's a catch. Oh, beautiful vase, yeah. Good catch. It can be a bit of a delicate sponge, that one. So
Apparently this one is quite delicate. It's hard to grab.
Yeah, hard to grab without smashing it. Yeah. Let's does the robotic arm have feedback sensors on it?
No, it doesn't.
Mm.
I think we're looking at another one of the cameras and we can see the sponge slowly falling down the slope, but the arm is there waiting to hopefully catch it.
That's all right. Yeah, glass sponges are funny because sometimes they're hard and super fragile like a champagne flute, and other times they're woven like a sheet of fiberglass, if anyone's worked with fiberglass, and it's all soft but really quite strong. So they're quite variable. So some are very hard to pick up. Plucking champagne flutes off the bottom. That's why I asked whether you had sort of a pressure sensor on the arms or not.
I think that one is a little bit too hard to grab, and the ROV pilot thinks it might be really, really dead, so it's quite fragile.
Well, even some of the live ones are fragile, but that's all right. You can look out for another one of those. As you can imagine, big glass sponges like that have been more likely to be caught over the last couple hundred years by dredges and so forth, whereas the super tiny carnivorous sponges like you saw hanging off the wall before, either overlooked or washed through big nets that were dragged along the bottom. So that's why they're new.
Yeah, it's funny, speaking of all these glass sponges we've been handling, because we've collected quite a few big ones, spiky ones, and Joan and I never really know, how they feel. So, we've held a couple of them and realized that, we've had little splinters from them, or some of them just crumble within our fingers because you look at them, but it's hard to guess how fragile they are or how sturdy they are some of the time.
Yeah, that's right. Same as shallow water sponges. And you could use that for quite a few IDs to help. Is actually give it the poke test underwater, you see what it feels like, and then on top, back in the lab when we're trying to identify it, actually, squeezing them and trying to rip them apart also becomes part of a description of a sponge because they're very different in the way they feel and behave. So it's not as much. It's a very non-mathematical part of description, those ones.
Yeah, if anything, it's the fun part of trying to describe them, to be able to feel them with your own hands and, yeah, as you said, rip them apart or squeeze them. And we've had to, cut off small pieces and fit them into vials. And, even through just doing that, you can feel the crunch sometimes between your fingers. And that kind of description is really only something that you can do when you hold it with your own hands.
Now we're looking at a very pretty gastropod here.
So apparently we have a permit to collect these gastropods, so if it's possible, we're going to try to collect this. If it doesn't roll far from us or roll down the hill.
Specifically asked for it.
So apparently there's a scientist called Dr. Patrick from Belgium, who specifically asked for this kind of sample, so a top snail. So yeah, this is actually ideal. We weren't expecting this.
But somehow I think today we have our avalanche meter on. This is definitely my first time seeing a snail moving that fast. It has really strong upper body strength just to push himself out of the way.
Yeah, he really has moved very far, just in that, those few seconds of not being able to see much. I think he's managed to escape. Oh, that looks like it might be it right there. We got him! Hopefully. Fingers crossed.
It's a very delicate grab of this gastropod. Very good job. So we saw this question from Clint on YouTube. So about the permits, since the Great Barrier Reef Marine Park is a really big authorization, and this is a very well protected area, depends on the research need. We usually will apply for permits according to our trip target or with the science team here we have coral specialists, we have invertebrate specialists. So all marine lives are equal, but it's just depends on each trip. We have a different target, so that's why during this trip we're mainly focusing on coral and sponges, and if necessary, this type of gastropod that are closely associated with the coral reef or deep water. So the gastropod we just collected is a type of top shell, and hopefully our onshore scientist Patrick from Belgium, he will be very happy for this species that we just collected.
So as Rob just explained to us, it's really interesting to look at these rock formations, because just by looking at the color, you can see right in front of us, the lighter colors are where it hasn't been as exposed, and so it doesn't have this layer of Veronese oxide on them.
Ferromagnesium.
Yeah, ferromagnesium oxide on them. So you can tell that a layer has fallen off and has shown this less exposed part, which is why it's a different color and lighter. Oh, it looks like to the left there's some more sponges. I don't know if these are similar to the one we collected, that lollipop-looking one in the beginning.
Can't quite see it, but if you can grab it, that'll be amazing. Yes.
Yep, we can do that right now.
Sure,
There's a few of
fantastic.
them down.
Thank you. Yeah, a few there. Fantastic.
So Merrick, is this type of sponge we collected a type of glass sponge since they don't really have the tentacles, or also carnivorous sponge has another type of appearance?
Yeah, that's right. There are glass sponges and there are carnivorous sponge that both have this sort of tulip-like shape. Yeah, and it has a whole lot of these anchor-shaped spicules inside the tulip-shaped thing that amphipods and isopods have a whole lot of hairy appendages, and they all get caught. Little hairy appendages get caught by 1, 2, 3, and then by the time they're caught by 20 or so of the anchor-shaped spicules, then it's trapped, and that's when the sponge can start digesting it. So, yeah, so that shape can be a glass sponge or a carnivorous sponge. It'll be really, when I look under the microscope, I'll be able to tell. So great work, guys. You're great at plucking daisies and tulips.
This is some amazing control plus luck. Just let it free fall to the boxes that we want to. Sorry, the wrong word. So apparently this is the business skill that our ROV pilots are referring to.
Okay, so someone just asked if there's an ambient hydrophone on SuBastian, and the answer is no, there is not. Exactly.
Oh,
I
such
think
good
everyone.
work, guys.
Yeah, thank you guys. I think if we did have a hydrophone, you know, all the noises that you guys were hearing today, complaining about on the live stream, you'd probably hear something like that.
Great work, guys, in the ROV. I particularly love the way you just quickly pluck it so it grabs the basal attachments. It's a fantastic job. I assume the HD camera's rolling at the same time as well, isn't it?
Yes, it is always rolling, so we have great footage of all of this. It's so useful having everything shot in 4K, but also sometimes going through it just means, I know that on my laptop it gets quite slow when I'm trying to watch everything in such good quality, so it makes it a little bit difficult, but at the same time it's so useful, you know, especially if we're describing a new undescribed species.
Great.
Okay, I think we'll
Great
probably move
job.
on upwards a little bit. Yeah, thank you guys. We'll probably move on a little bit. I think we're trying to move as quickly as possible, and we're you know, making our way upwards. It's just so tempting to stop all the time.
Especially when this is the only chance we get.
These look like very beautiful sponges. Any of these of interest? Oh.
Yeah, grab the whole rock if you like.
Oh.
Just what's often grab the rock and you'll get at least 2 sponges and a rock sample.
Yeah, true. Win-win. We can please everyone in the room. Now let's see if this rock is going to be a pain to remove or if it's going to slip out easily. I highly doubt it, but, you know, fingers crossed. We've been lucky in the past.
There we go.
Oh, that's
Sitting on the
looking
floor.
perfect, yeah. That's amazing. Thank you, guys. Everyone will be very happy with this. Yeah, I have been talking about how we're pleasing everyone. We do have to mention, though, we are not collecting as many black corals as obviously we would want to. You know, Joan and I, that's our job, so we should also be looking out for those because, you know, we have to try and please everyone on this dive. Including you, Jeremy. Shout out.
We would really all agree that just now was a really good catch of this trying to escape sponge.
Was a good job. Yeah, that sponge it's a glass sponge. It's possibly one called fire ochre. Yeah, what's really interesting about it, last time we got one from Osprey once recently, and it has a whole lot of super tiny epizoanthers, which are going to be a new species as well on it. But until we look at it under the microscope, I can't tell what species of sponge it is. And then we'll have to look at the zoanthid if it's on there, and see if that's a new species too. It's all pretty exciting.
Now we are seeing at the bottom, it's a headless chicken monster. It's a deep sea sea cucumber. The color is really nice. Now it's trying to swim upwards in the water column. And I think the pilot developed a habit from my story yesterday, trying to look at a specific part of the sea cucumber.
it does have this beautiful color, and it's funny to think because at first I thought, you know, looking for a Dumbo octopus everywhere, I thought it may have been a Dumbo octopus, but it has similar shape. But you know, when you look at this, you really don't. Well, me personally, I don't really think it's very cute at all, and I think having eyes really makes the whole difference to differentiate what is cute and what is not.
Exactly, especially thinking about sea cucumbers' diets, they are usually feeding on sand, which they're trying to extract those organic nutrients from the sand, make it available in food web again. And poop they're having are basically just clean sand, so they are actually very important to the ecosystem, just to recycle the nutrient and clean the sand. And usually when we're saying in a shallow reef, a sea cucumber can help to reduce the nutrient amount in the water column that can help the growth of corals and others like seagrasses that requires relatively lower nutrient level dissolved in the water.
Yeah, close up that
Looks
one.
like another sponge
Same
in
we
front
had
of
before.
us. Yeah, it looks like a big tulip one.
What about the small little white spots to the right of it?
Is this the one we're currently looking at, or?
Yeah. Closer up. For, no, I think it's just not worth grabbing. Thank you. Let's see what's further under that overhang there. Yeah, it's all right. See that whip that's hanging down? Oh, sorry, it's zoomed out now.
So is it the like a whip shape with some spikes
Yeah,
on
have a look
top?
at the whip one. Does it have? Yeah, does it have a whole lot of filaments? All at right angles to it?
We're zooming in and trying to focus right now.
Thank you. Yeah, that looks good. That looks like a carnivorous sponge. If you possibly grab that one for me, please. Oh, can you see the tentacles? If it's got tentacles, it's not a sponge, but if it is fine filaments, it would be a sponge.
I think it looks like fine filaments. But yeah, we are
Yeah,
collecting it now.
Yeah, it's probably quicker to grab it than spend 10 minutes trying to focus on it. Very exciting.
just answer a couple of questions on the YouTube channel. Currently, we think this is a sponge, since it's having very fine filaments that doesn't look like a coral tissue covering on top of a skeleton. But since we are trying to climb up the hill in a relatively short time, so we will have a closer look after we take it back to the lab.
Yeah, it does. It looks like a carnivorous sponge to me. They don't look like other sponges at all. These ones are soft filaments. It's a soft like thread, and it's got these super tiny filaments out the side, which actually help stabilize it and also catch prey. And yet again, it'll possibly be a new species, quite likely. So, very exciting when it gets back to the lab.
I feel like Falkor's logbook will be like, new species, second new species, third new species, and just keep going.
Yeah, that's right. That's
We actually need a new species-ometer. I think that's the exciting meter that we haven't had yet. That would be off the charts, I think, at this point.
Yeah, that'd be great. The only problem is it probably takes about 2 years to prove it. Not as fast as other groups like fish. It's much faster to see if it's a new species, isn't it?
The harsh reality of science.
Yeah, that's right. And yeah, some bits of science move faster. And you were talking earlier about sponges moving between groups, and I love a quote, and I can't remember whether it's Marconi, I think, might have said it, and it was science moves one funeral at a time. Sometimes you have to wait till someone's passed on before we're allowed to get rid of that theory, and they won't argue about it stressfully, and everyone will accept the new theory. So it's faster than that, though, this one.
Yeah, that's a grim thought, but that is true. We have to keep powering on through it. I think it's good that we have so many people who are so passionate, because otherwise it would be hard to know that, you know, a lot of the things that you do in your lifetime, you have to wait for, as you said, a funeral.
We're looking at another sponge right now. It's like a really big body, but attached onto a very thin stem or stalk-like structure. This usually be a strategy for them to extend out and feed in the water column. Yeah,
that's exactly right. They'll have the right. This is a glass sponge, and so this is a filter feeder. But they'll have exactly the right amount of they want to get where the current is just at the right level for them. You know, strong enough to be bringing nutrients in, and probably not too strongly to blow them away, but they want to keep away from the sediment layer and the still water layer, the laminar effect. Yeah.
Do you think this one is worth collecting, or we had something similar already?
Oh, I think we have something similar, but it's an easy one to grab. Why don't we grab it? You can compare it to the other ones easily. It looks a lot bigger than the other ones.
Yeah, we're starting to realize that we have our boxes filling up, because we're just trying to figure out which box to put what in, because we've got plenty, and I think we're all getting really excited.
And the challenge later on is sorting them all out. At least have lots of little boxes rather than one big net that ends up with 2 tons worth of stuff.
Yeah, exactly. And we usually have it all separated in different boxes, so it's not as difficult. And in our notes where we're logging, we actually write a little description, so it isn't too hard for us, but that's something we have to look forward to after this dive.
Well, thank you very much, guys, for sorting that on, doing the on-deck sort too. It's great. Sorry I couldn't be there to help you out with it.
Oh no, of course, it's our pleasure. If anything, I know that, you know, especially for me and Joan as students, we're so lucky to be here. So if anything, you know, we'd be happy to do anything.
It's always great, and because you're so deep here, and this stuff is, you know, brand new to science, no one ever gets there, so it's fantastic to be able to look at it and touch it and see it. And I still remember the first time we went and did some deep water stuff too. It's all like, oh, I don't know, it's all new. So it's really good.
Yeah, exactly.
Good learning experience.
That's pretty much exactly how me and Joan react every single time we touch something new. And we get, yesterday actually, we collected some very slimy black corals, as we mentioned on the live stream, and touching them was even more slimy than we could have expected. And we were washing our hands several times, nothing was coming off. You know, we didn't even want to go eat dinner because it was just sticky everywhere. And as I've said with the sponges as well, you know, we've come back from the lab with little spines in our fingers, and it's just been, you know, an exciting time in the lab every single time, because we have no idea what we have to expect.
Yeah, it's one thing about sponges, you inevitably end up with little spicules in your fingers. There is one of the sponges in shallow water, like as in 30 meters, this black sponge Iotrocia. And every time you pick it up, you don't notice it, but about a week later, all the top layer of your skin all peels off.
Okay, I'm guessing that's only for shallow sponges, and we don't have to expect that for ourselves.
Not that we know of, no.
Okay, noted, that's good to know. What about this sponge in front of us?
Yeah, look, we can stick it room in the bio box still. It's a beautiful glass vase. Yeah. Oh, look, it's great. You can see all the spicule filaments on the outside. That's the sort of thing that never gets captured before either. You never see that sort of thing. That structure is always breaking off. So, again, another first for SuBastian. Great work.
So we're actually thinking of maybe flash freezing this one because... Oh, the coral next to it, okay. We've got, yeah, this sponge that we're going to collect, and we're also going to collect the octocoral on the side for Jeremy,
Yeah.
so it's a double win again.
Yeah, this sponge is probably a Euplectus species, and it possibly has two shrimp in it, a male and a female. So I think they are. And when the sponge is young, the shrimp can go inside, and the sponge sort of grows around them like a net, and they're trapped in there for their whole life. And Japanese people use this as a wedding present because it symbolises marriage, as we'll be together. See anymore? Anyway, they should survive, hopefully. Fantastic vision, guys. Well done. computer.
No, so I think we're actually going to skip this coral because we actually were thinking about flash freezing the sponge, but unfortunately it was a bit too big for the quiver, so I think we're going to move on. We're just having a discussion back here in the control room because we're looking at everything that we have in the bio boxes, and I think we still have just about 45 minutes left to this dive approximately, maybe a little bit more, but we're worrying that we have to start focusing really on our priorities to sample, because a few of the boxes we can't actually open because there's a lot of floating samples in there and we wouldn't want to lose everything. So yeah, I think we're gonna try and get the most important ones right now, if possible, for everyone on shore in Merrick.
Yeah, well, that's good. Don't worry about the big glass sponges or the big sponges. I'm much more interested in the tiny ones myself. Little carnivorous ones. Yay!
I think what we were just seeing now is a little scale worm that was swimming in the water column. And there was another snail, a similar one to the one I think we collected.
Yeah, top snail here, a top shell.
This one's a lot less frantic than the other one that we saw that was jumping around everywhere and trying to escape us. He looks a lot more relaxed.
I think it's getting late and they are getting a little bit tired as well. And right now we are at the depth of 1800 meter below, and the temperature is only around 2.4 degrees. This is really cold. I can never imagine myself swimming in water at this temperature.
No, neither. I already start to get cold at about 28 degrees Celsius, so I can't really imagine. Well, I mean, I have gone, you know, swimming in maybe sub 10, but yeah, as I said, even below 28 degrees Celsius, I start to complain. So 2.4, I would definitely not get in the water. I've probably only just done it once, I think, in Iceland. Went diving between two tectonic plates in the Silfra fissure, but yeah, that was ridiculously cold, and is one of those things that was amazing to do once, but I'm not sure if I would do it again. This looks like a prime avalanche zone, so you can see a lot of things like have left trails while falling. What that
So
looks
here
like
we have something hanging on the rock, looks like a siphonophore. This orange mech or swimmer structure on top.
a benthic. Is it something that maybe Dhugal would be interested in?
If he's still here with us.
I know in one of the previous dives he was looking out for a benthic siphonophore, so not sure if this is of interest.
We have no sample jars for that for sure. Apparently we tried to sample this benthic siphonophores before, but they are a bit extra sensitive, so once we get too close, they might just spit all of their guts out and make it quite difficult for us to do anything else.
Good to just admire from afar, I guess, and get some nice pictures or video footage of it.
Yeah, that looks like the Falkor we collected just before. I can't see any of the zoanthid on it we're looking for.
So,
Should be all
Merrick,
poking out like little flowers.
I have a question actually. In the center, you can see there's a slight pink tinge to the sponge. Is that because there's a worm inside, or yeah, what could that be?
Yeah, you're right. It could be possibly anything inside there. Like I said before, there's sponge hotels, particularly this one, which is like a whole series of cylinders. So anything small enough can wander in there and stay in there. Could be a very small lobster. Could be anything. Yeah, a worm. Yeah. So it's not the sponge, but one of its hotel clients, if you like. Beautiful photo, close up though. Fantastic, guys. Let's see what it looks like. Isopods sitting out the side. Yeah.
I think we have more sponges here. The one on the left looks quite interesting.
It looks like the same species as before and got the same patrons of the hotel as well.
Yeah, exactly. Look like little squat lobsters or something residing in them.
You can see we're really hitting a great depth and obviously current for these two particular sponges, these big tulip sponges and the Falkor one. So they're obviously got their niche right there.
Yeah, we've managed to see quite a few of them in different shapes and sizes scattered around. I think we managed to sample quite a few, so it'll be good to take a closer look at them later as well.
Got a really
Absolutely.
colorful sea lily that is coming up.
Oh, nice.
It's a beautiful color.
You're not going to collect one?
Unfortunately, we don't have a specialist on this yet.
I think there'd be quite a few people who would be interested in that. You can collect it, yeah.
Yeah, I think unfortunately we don't have much space, even if we did have the permits.
Yeah, fair enough. We had exactly the same effect over an Osprey reef many years ago with an ROV, and we collected two of these, an orange one, a yellow one, and we went back to get a third thing, and as we picked it up and opened the case, the two crinoids floated off into space, never to be seen again, and we never found them ever again.
And apparently that sea lily just thought our camera lens might be a proper food to grab.
Oh yeah, this is a very nice rock. Nice little cave below it. Another one of those snails rolling around. We've got another bamboo coral here, similar to the one we collected earlier with little squat lobsters on them. Yep, so we're gonna collect this sample again. So this bamboo coral that we're collecting is for the full genome project, so we will be flash freezing this one in liquid nitrogen. Yeah, it's funny to think this will be the last time that we'll be opening the doer and flash freezing all our samples today. A little bit sad about that.
I know, I will miss that a little bit for sure, thinking how cold those liquid nitrogens are, and we need to put 2 gloves onto those metal rods in order to pull them out, and it is quite a tricky design to pull those spoon out, but after a while, when we get used to it, it is still very cool to see all those condensed air and water molecules plus nitrogen is coming out of that big jeweler.
Yeah, I know that Joan, you've worked with liquid nitrogen before, but I had never previously, so this was actually my first time, I think. I don't know if we'll be there to witness them pouring it all out, but I hope that we are, back when we return to Townsville and we meet up with Jeremy Horowitz and Tom Bridge back at JCU. So hopefully we'll be able to witness that and see, you know, our little piece of equipment that we've been using for the past month, and yeah. Hopefully we will get to witness the end of it.
We're just heading this back to Marty and Rob again.
Hey Merrick, you're still online?
Yeah, I am. I'm saying that little yellow sponge past the robotic arm there. Just have a quick look at that. Yeah. Is that what you got to point me to, Rob? Or?
Oh yeah, just having a look. Just Marty and I are settling back in the audio chair here. Oh, it's been a big day.
Thank you very much to Valerie and Joan for looking after the commentary for a little while we were busy doing other things on board the ship, and also to Vicky for chatting on the commentary a bit earlier on today.
All right, so we are really at the tail end here. We're just going to roll up the slope for a little while longer, but not much longer, perhaps for the next 5 minutes or so, and then we'll call it quits. There's one of these top snails doing this weird dance over the mud. But we've got some samples of this that'll keep Patrick happy. Patrick's I forget his last name, but it's he's been interested in these right from the beginning when we first started looking at the Coral Sea, and he would send me these very grainy YouTube snapshots and say, please can I get one of these? And anyway, eventually we managed to get a permit for him, so here we are. We've got a couple of samples of his top snails, so I think they'll end up with you, Merrick, and then we'll have to coordinate to get them to Belgium, but I'll give you the details of that later on.
No worries.
So look, we're just going to crank it on up. We should get some music going. Some Texan music. That's what we really need. Got the Texan crew here. We've got J-Rod, Cody, of course Chris is part of the team. We've had John and James on Wirecast throughout the dive. Marty, it's been a real pleasure on this dive. We wanted steep and deep, and didn't we get it?
We certainly got it, didn't we? How good was that? At the bottom of that waterfall, those boulders Yeah, we didn't know what we were going to find. It could have just been a pile of mud. But
I was
boy,
expecting a
it
pile of mud.
disappointed it.
Steep mud, I was expecting steep mud, but nothing quite as spectacular as this, you know, just jumbled piles of rocks. And, you know, I'm a marine geologist, and I loved, you know, as an undergraduate, I've walked over many a sedimentary rock environment, and it's just incredible to see it down here 2 kilometres deep.
Yeah, that's right. Well, we'll actually be going back over this video footage many times, I would imagine, looking at all these different rocks and the stratigraphy, the layering that we could see. So even though we weren't in the control room for a while, there are screens all over the ship. So we were certainly watching.
Yeah, you know, and I do love those quiet moments where you just zoom in on a stalked lily. You know, I think they're amazing. They are a relict animal, and here they are in the deep Great Barrier Reef, just sitting there quietly. You know, they're just amazing animals.
In the inset there, you can see our dive track today, from where we started off at the bottom of the plunge pool, and we've made some pretty good progress up the side wall of the plunge pool.
Yeah, pretty good progress, but we're still only halfway up. We're halfway
through what we planned.
Yeah, but we had the pleasure of Dhugal Lindsay joining us for a midwater dive, so it's quite a lot to combine all together. Oh boy, I do like looking at these layers here. But you know, we do have to say good night and goodbye, farewell, au revoir, Wiedersehen. Because it's getting on here on the Falkor. We've been diving all day. It's going to take a good hour to recover on board, and if we don't get going soon, it'll be night time, and we've still got lots of work to do. In fact, the ROV SuBastian is being packed away. This is its last dive.
Yep, this is the last dive, not only for our expedition, but this is the last ROV dive for Schmidt Ocean Institute and RV Falkor for the year.
That's right, in Australian waters. So we'll be packed up over the next few days as we head down towards Cairns, which is the end of our leg 2. The voyage continues. It'll be leg 3, but it'll be mainly focused on a multi-beam mapping exercise. There'll still be outreach. We'll still be writing blogs. Carly Weiner, our hard-working Director of Outreach and Communications, will be getting her team to help continue put out outreach. So the expedition isn't over. We just won't have the ROV with us. The ROV team is heading home for well-earned break. But gee, it's been just a fantastic ride.
Yeah, it really has. I mean, when we set out from Brisbane 4 weeks or so ago now.
How long ago was that?
How long ago was that? Yeah, with, I don't know, an open mind as to what we might discover and uncover, and it's certainly just, I guess, been well and above, beyond any expectations. It's been really fantastic, the things that we've mapped and discovered and uncovered, whether it's the geology or the biology or the seafloor features that we've mapped. It's just really been quite extraordinary, and an absolute pleasure and a thrill to be a part of something like this. It's so significant.
Yeah, that's right. It has been a pleasure, and to see everyone in action here. It's not just the control room, but the entire Falkor team, both ashore and on board. We have about, I think, 26 or so full, crew members. The stewards, the chefs, the engineers, of course, the bridge team working 24/7. It's an amazing team effort. It's just wonderful to be on board and just see everyone working together, in support of us. We feel very much like a part of the family here, and I guess that sort of brings me into the chat family. All of those familiar names and handles that we've seen over all of these dives. We don't know your faces, but you do feel very close. You become very familiar. The comments, we really appreciate the humour that helps us, and also the commentary, the audio from the other scientists who are joining us. But yeah, I think overall what I've learned from listening and also reading all the different chats, that's been fantastic, hasn't it? The people ashore.
Yeah, absolutely. I mean, we think of them as armchair experts, but there's really has been a lot of expertise coming through on the YouTube chat. There's only a small science team on board, and we certainly don't have breadth of expertise across all of the disciplines that you can experience on these expeditions and ROV dives. So really having that contribution from onshore, whether it's our scientific colleagues or everybody at home onshore that joins in and chats with us, and a lot of expertise there as well, which is really, really fantastic.
Yeah, thanks everyone. Great to be joining it, even though I've been such a long way away. So thanks for all your great work on board. Great work, guys.
Yeah, thanks, Merrick. Okay, well, we'll finish it up here. So I'll just say this is Dr. Robin Beam from James Cook University saying farewell from the far northern Great Barrier Reef, offshore Cape York area. This is the end of Falkor Dive 402, and from here we are heading back to Cairns. So say good night. Good night, everybody.